Macromolecules of Life by Zuhair Hassan and Milagros Santos. Amino acids, fatty acids, monosaccharides, and nucleotides are important molecules. These four molecules are the basic building blocks of the macromolecules used to construct our cells. There are four types of macromolecules found within all our cells: carbohydrates, lipids, proteins, and nucleic acids. Amino acids are also important macromolecules, but was not tested for in our experiment in lab. These macromolecules organize into a basic functional unit we call our cell. Carbohydrates. Carbohydrates are organic molecules composed of carbon, hydrogen, and oxygen atoms. Sugars are a type of carbohydrate, and the simplest of these sugars are called monosaccharides. For example, glucose. Once glucose enters the cell, enzymes can break it down into smaller molecules. Then, energy found between the chemical bonds in glucose is released and stored in the bonds of ATP. ATP can then be used for energy and various cell activities. The term monosaccharide translates into single sugar. Sugar is used as a source of energy in living organisms. There are three types of carbohydrates: monosaccharides, disaccharides, polysaccharides. Monosaccharides are single sugars. These are also called simple sugars. We tested for simple sugars and complex sugars in our experiment. Complex sugars are polysaccharides. For this experiment, we used Benedict's solution, 4 test tubes, distilled water, a sample of each substance, glucose, starch, egg, vegetable oil, and 2 other unknown substances in a beaker of water and boiling chips. Distilled water was needed to be boiled at a temperature at about 40 to 50 degrees. Next, the 6 tubes were prepared with each sample substance. 40 drops of each substance was added to 1 tube. The rest was done for the rest of the tubes. After that, 400 drops of Benedict's solution was added to each test tube. 40 drops. 40 drops of Benedict's solution was added to each test tube containing the substance. After that, all 6 tubes were mixed evenly. Then the tubes were sitting in the boiling water for approximately 5 minutes. If the solution turned green, yellow, or brick red, then it contained carbohydrates. Once it was mixed, all 6 test tubes were put back in the water and were cooled for 5 minutes. If the solution turned green, yellow, or brick red, then it contained simple carbohydrates. The positive control in this experiment was glucose, and the negative control was distilled water. Next, we will show a video of our results. This first test tube was our positive control, and it contained glucose, and once it reacted with the Benedict's reagent, it turned this orangish color. So we can immediately tell that this is positive for simple carbohydrates. This one had water, and it didn't react with the Benedict's reagent. It didn't turn that orange color, so we can tell that it has no simple carbohydrates in it. This one contains starch, and it did react with the Benedict's reagent, turning a light orangish color, meaning that it did have carbohydrates. Next, we tested for complex carbohydrates. Complex carbohydrates are called polysaccharides. Polysaccharides are more than 3 monosaccharides linked together. These are known as complex sugars. An example of a polysaccharide is starch and glycogen. Starch is found in plant cells, and glycogen is found in animal cells. These 2 polysaccharides are used to store energy in our cells. Other polysaccharides like cellulose found in plant cells help with providing structure instead of storing energy. For this experiment, we used glucose solution, 4 test tubes, sample of each substance, and a plastic dropper.
Test for complex carbohydrates, we're gonna use glucose solution. This is required for this experiment. This will help to detect starch. We have different solutions. We have 1, 2, 3, 4, and 6 are unknown solutions. Number 5 is H2O, and number 7 would be the starch.
5 is negative control, and
7 is our positive control. So we're gonna add 10 drops of iodine glucose solution to each of these tubes. And see if it have any changes in it. You can see it's just mixing. It changed to brown, that light brown color. Look like dark blue. Like an orange, dark orange. Orange. Blue or black, then we'll know that it contains starch and therefore it's a complex carbohydrate. So far, number 2, our unknown substance turned completely dark. And number 7, which is starch, react with other starch. Not solution, so it'll turn black too. So in this case, our negative control would be unknown number 2. And
our negative control is 5.
Oh yeah, negative, right. The other one positive, yeah.
Lipids. Lipids are hydrophobic molecules. The main characteristic of all lipids is that they are nonpolar, making them insoluble in water. There are various types of lipids. These include fats, phospholipids, and steroids. Fats mainly function in storing energy. Hydrophobic molecules are made of hydrogen and carbon atoms. Saturated fats are mostly found in animal products such as meats and some dairy products. Unsaturated fats exist in nature and have a high melting point. The materials we used for this experiment are dropper bottles of distilled water and vegetable oil, 6 test tubes in a test tube rack, stock bottles of hamburger juice, onion juice, colorless non-diet soft drink, and cream, dropper bottle of Sudan reagent, vortex mixture, piece of uncoated paper. Here we have the 6 test tubes. 3 milliliters of H2O, distilled H2O, was added first to each tube, and then 3 milliliters we are adding of each substance to the test tube. Later on, 9 test tubes of Sudan reagent will be added to each tube, and the tubes will be mixed. And then
we are testing, we're using
later on, 2 milliliters of distilled water will be added to each tube.
To identify the presence of fats and oils. We are adding 9 drops of this solution into our different test tube.
Sudan V.
The first
Sudan 5.
4 and number 6 are unknown solutions, and number 7 is vegetable oil, and we're also using number 8 is the cream. So let's see how this goes. Yeah, it looks like the positive results are number 7, 8, and let's see, maybe number 6. It doesn't look like none of them knows have fats on it, except for number 3, or maybe number 6. Don't miss this oil. Should be go to the top. Yeah, that's okay.
Proteins. Proteins are composed of polypeptides which are arranged in a sequence of amino acids. Proteins do everything in our cells. The amino acid chains are nonpolar, polar, uncharged, and polar charged. Proteins assist DNA and RNA synthesis and regulate gene expression. They act as motor proteins, serve as defense proteins, increase rates of chemical reactions, enable our cells to communicate with other cells and with the environment. They also support and strengthen structures and promote the movement of solutes across plasma membranes. There are three structures of proteins: primary structure, secondary structure, and tertiary structure. For our experiment, burette reagent was used to test the presence of peptide bonds. The greater number of peptide bonds, the more intense the reaction was with the burette. Materials we used were 5 test tube racks labeled correctly with China market, stock bottle of burette reagent, distilled water, starch solution, egg albumin, glucose solution, and a colorless soft drink. For our procedure, the tubes were labeled 1 through 5, and then 10 milliliters of the stock solution was added into each test tube. Then 10 drops of burette reagent was added to each tube, and it was shaken and mixed thoroughly. Finally, after 2 minutes, the data was recorded. This video will show you what occurred. If the reaction turns a darker color, the darker color of the reaction is the positive control. How much of this should I put in now? Do you know what color it is? Water, substance. I think it's okay if we talk, so we're gonna like read that. Yeah, why? Number 4?
Mm-hmm. 4 and 6 are.
We still have to see if they're positive. Where's 4? 4 right there. Purple color. Amino acids is one of the things we did not test for in our experiment. However, they are polymers, which are relatively strong acids found in the nuclei of cells. DNA and RNA contain the genetic codes to make RNA and contain codes for the primary sequence of amino acids to make proteins.